Welcome back to another lecture video. In this chapter, we are going over the metabolic pathways for carbohydrates. And what we're really going to look at here is the first part of sugar digestion, really. We're going to see how it gets broken down. In the next chapter, we'll actually look at how we get energy from those broken down products. But in this chapter, we're just going to look at the various ways that glucose and other sugar molecules get broken down for energy, at least for the first in the first process. So we'll start with just defining metabolism. It's actually two processes. There's the catabolic reactions, which are the ones that break down molecules. So catabolism is about breaking molecules down. So those would be your sugars, your fats, and your proteins. And then there are the anabolic reactions. This is the other side of metabolism. This is the building up of molecules. And so when we store sugar as glycogen, or if we store triglycerides as fat, all of these things would be anabolic. And these typically cost energy, where catabolic reactions typically release energy. So we're going to start by looking at catabolic reactions in this chapter. And there's a few different stages. So stage one of a catabolic reaction would be the breakdown, essentially, digestion and hydrolysis of large molecules that can be made into small ones that can then get into the bloodstream and transported around the body. Now, once we can get them into the cells, when they're small enough, we break these down into, so these larger molecules, into 2 and 3 carbon compounds. And we're going to name some of these compounds here in a few minutes. And then stage 3 would be oxidation of these molecules. And this goes through the citric acid cycle and the electron transport chain to actually turn that energy into ATP, which is something that we could use. Now, there is a lot of energy also being released as heat, and we'll talk about that towards the end. So in the 3 stages of catabolism, large molecules from foods are digested and degraded to provide smaller molecules that can be oxidized to produce energy. Okay. And so you can kind of see here the 3 different places where those things are happening. Okay. Well, actually, I don't know if you see the places necessarily. No, I guess not. So let's just say stage 1 doesn't happen in the cell, but it's to get molecules small enough to get into the cells. And then the inside the cell, there are 2 main objectives. One is to break the molecules down smaller, and then to release energy. Now, in the cells of plants and animals, these are known as eukaryotic cells. And we haven't gone a lot into this. This is something that you probably go over more in biology. Eukaryotic cells are differentiated from prokaryotic cells in the fact that they contain a nucleus, and that's where their DNA is kept. Now, a nucleus is characteristic of eukaryotic cells. There's actually a few other criteria, but that's the main one. And so the cell membrane separates the materials inside the cell from the aqueous environment outside the cell. Within the cell environment, we have lots of different organelles that do different jobs. We've talked about the ribosomes and their function in protein translation. Lysosomes are molecules, or they contribute to the breakdown again of either molecules, or they have oxidizing agents that are stored inside them. Now, the nucleus, we know, contains the genes that control DNA replication and protein synthesis. And the part kind of shown here, these varying membranes coming off of the nucleus, the rough and smooth endoplasmic reticulum, are all just part of packaging and sort of moving proteins out of the nucleus. But the cytosol and the mitochondria are what we're going to be focusing on. So the cytosol is the fluid part of the cell. It contains electrolytes and enzymes that catalyze many of these reactions that we're going to be talking about. And then, of course, there's the mitochondria. The mitochondria are what we call the powerhouses. This is where ATP is produced, and it's produced inside the matrix of the mitochondria. So when we start to get into, I guess, the next chapter, right? In this chapter, we are going to be looking at how sugars get into the cell, how they're broken down into those 2 and 3 carbon compounds, and then I think in the next chapter we'll look at how, actually no, we'll look at a little bit of those intermediate products, the electron carriers here, but in the next chapter we'll take a trip inside the mitochondria so that we can talk about ATP production. So that'll be the next chapter. But before we can even get in here, we have to get our molecules pretty small. Okay, and then here's just a little sort of function description of some of these most important organelles or cell components. So one of the molecules we want to talk about here is ATP. Now, not just because this is the energy sort of currency of the cell, but because it is involved in a lot of different aspects of metabolism. Now, the ATP molecule is composed of the base adenine, which is where the A comes from, a ribose, sugar, and 3 phosphate groups. They hydrolyze to form ADP and AMP if we were to remove those phosphate groups. So ATP has all 3, ADP only has 2, and AMP would only have 1. Every time that we hydrolyze one of these bonds and and free up a phosphate group, energy is also released. So this is how we pay for things with ATP molecules. It has stored up energy in it. There's actually a number we can put to that stored up energy. The hydrolysis of ATP to ADP would release about 7.3 kilocalories, or 31 kilojoules, depending on what unit that you are looking at, per mole of ATP. And it is the same for ADP to AMP. From adenosine diphosphate into the monophosphate, we would liberate another round of 7.3 kilocalories. Now, the point of releasing the energy by hydrolyzing ATP is to drive other reactions that normally require energy for them to happen. So this is a spontaneous reaction that can drive non-spontaneous reactions. And so that's the point of it. And you can see here, if we look at ATP, and we know that ATP releases energy. Now we look at this other reaction that we want to make happen. We want to take glucose, and we want to be able to basically use it for energy. We want to oxidize it. In order to do that, we have to sort of prep it for the reactions that it's going to go through. The prepping of glucose costs energy. It costs about 3.3 kilocalories. But because we can release 7.3 kilocalories every time we hydrolyze an ATP, that more than pays for the cost of that other activation. So by coupling reactions together, this is how your body is able to build things. It uses the energy from breaking things down. So quick learning check. Try to match these up. So 1 is ATP, 2 is adenosine diphosphate, and a free phosphate. So it would be pre-hydrolyzed and hydrolyzed. So see if you can figure out what goes with what. Pause it, give it a shot. Used in energy requiring reactions, ATP. The energy storage molecule, ATP. Combined with energy requiring reactions, ATP. The hydrolysis product, ADP. Hopefully that wasn't confusing. It's meant to be straightforward. I hope you didn't overthink it too much. Now there are some other important coenzymes that we should talk about. Coenzyme A is one of them. NADH is another. FAD is another. NAD plus, instead of NADH. NADP plus. Anyways, we're going to go through these, and I'll show you all of them. But the goal here would be just to be able to kind of talk about the different components and functions of these various coenzymes. But again, coenzyme A is shown here. We'll talk about it a little more in a minute. So these metabolic reactions, they often involve lots of oxidation and reduction reactions. And of course, whenever we say oxidation, we also mean reduction because they have to happen at the same time. But they're enzymes that undergo the reactions, or that I guess carry out the reactions. They're often named for the job that's being done, and in most cases they're either um, um, dehydrogenases or oxidases, and we need to know how that relates to um, oxidation and reduction. So oxidation in our usage here usually involves either the loss of hydrogens or electrons. And again, oxidation, reduction, um, oil rig can help you. Oxidation is loss. So the loss of hydrogens, loss of electrons. Whereas the reduction would be the gain. Reduction is gain of hydrogens or electrons. But there's also a definition that involves oxygen. When you are oxidized, you've gained oxygens. You've increased the number of oxygens you have. When you're reduced, you have less oxygens. And so we're gonna see that in most cases, it's going to be the addition or removal of hydrogens that tend to kind of give us the names of these reactions. Now, in both types of these reactions, the coenzymes that we're going to be talking about, they're required to be sort of an intermediate between, let's say that something's being oxidized and something's being reduced, and it's hydrogens that are being removed and added. Well, one of these coenzymes is going to take the hydrogen from someone and then give it to someone else. And so the coenzyme itself will both be oxidized and reduced. That's why they're such versatile molecules. They can oxidize or reduce depending on what form they're in. So this should kind of help a little bit with the definition of reduction and oxidation. So one of the molecules that we're looking at is coenzyme NAD+. Now, NAD+ stands for the nicotinamide adenine dinucleotide. That's the acronym there, NAD. Plus comes from the fact that it is ionized, and kind of like our work with amines and our work with carboxylic acids, we know that in different conditions they can be ionized, right? We can have carboxylic acids or carboxylates. We can have amines or ammonium ions. So this is similar. So this is an important coenzyme. You can see kind of where it comes from. The vitamin niacin provides the nicotinamide group, so that's why we need that vitamin in order to make this molecule. There's a ribose sugar, and the adenine diphosphate, ADP, is actually part of this structure as well. It's very good at being an oxidizer. Or I'm sorry, it can do oxidation reduction. Redox is a very good redox molecule. All right, and one of the things that it's typically characterized for, it's required in reactions that produce carbon-oxygen double bonds. Carbon-oxygen double bonds. So it increases the bonding to oxygen. And so oxidation of alcohols to aldehydes and ketones would be something that NAD would be useful for, right? We know alcohols oxidize into aldehydes and ketones. NAD plus would be the coenzyme. So if this is being oxidized, then that means NAD plus has to be reduced because they have to happen at the same time. I guess what I should have put here was H2, right? Because we usually say reduction is hydrogen gas. So you can see here that the structure NAD actually reflects that reduction, NADH. I said structure, I meant formula. Let's look at the structure. So you can see here the oxidized form of NAD+. So NAD+ is everything up to here. Ignore this guy for a second. So we've got the adenine dinucleotide, or the adenine diphosphate guess we're not really seeing where the phosphates are here. We have the ribose. Oh, my bad. Adenine diphosphate is this bit right here. It's the nucleotide, the sugar, and the two phosphates. Then there's an additional ribose sugar. And then the nicotinamide section of this comes from the niacin vitamin. So just looking at this part right here, okay, that's NAD plus. Notice right here on the ring, it doesn't have any substitutions. The nitrogen has a plus charge on it because it has 4 bonds. Okay, when this thing gets turned into NADH, notice the position of the double bonds. They move, right? Because now we've added an extra H. I guess the pink one's the one we actually added, but it doesn't matter here. Here there's 1 H, but now we've added 2. That forces the double bond here to move. Moving the double bond now removes it from the nitrogen, and you can see that the nitrogen no longer has a charge. But it also has an H on it, so that's why we write it as NADH. And there's also a free hydrogen that goes floating around, which is why it's shown like that. It doesn't really float around. It's probably part of a water molecule. Okay. So that is coenzyme NAD plus and how it gets reduced. Now there's also coenzyme NADP plus, and it becomes NADPH. All of that stuff still happens. The only difference is that on the NAD side of it, on this 2' OH, there's a phosphate group. So that's the difference between coenzyme NAD and NADP. Okay, NADP just has a phosphate on it. We call it nicotinamide adenine dinucleotide phosphate. It's used in anabolic reactions such as lipid or fat synthesis and even nucleic acid synthesis. So we don't see it used as much in the catabolism as we do in the anabolism. It's very similar to NAD+. And again, only difference is that 2' OH group. Okay, so that's 2 of the enzymes. I'm sorry, that's 2. There's 2 more. There's FAD and there's acetyl-CoA. So let's take a look at those. So FAD, flavin adenine dinucleotide, contains an ADP, just like the last one did. So here's our ADP. It contains a riboflavin, vitamin B2. And so I guess that's all of this guy, riboflavin. Now, when this thing undergoes the reduction, 2 nitrogen atoms in the flavin part of the FAD coenzyme, they're probably highlighted here. These 2 react with some hydrogen atoms and electrons and become reduced. So they gain hydrogens. And so FAD reduces into FADH2 when you are looking at the formulas. So NAD plus into NADH, NADP plus into NADPH, and then FAD into FADH2. No charges on that one. All right, now FAD, I give you the name here in case you wanted to write it out. This one participates in reactions that produce carbon-carbon double bonds. So as opposed to the NAD plus being carbon-oxygen double bonds, sorry about all that notation there, or essentially oxidizing alcohols, right? This one does more reducing. So by forming carbon-carbon double bonds, we tend to lose some hydrogens. Actually, I think that's oxidizing as well. My bad. So both of these are oxidizers, but they're oxidizing in different ways. So forming carbon-carbon double bonds instead of carbon-oxygen double bonds. Um, there's an enzyme as well that helps to actually reduce this molecule, or in situations where this molecule is used. So this is a very specific reaction, part of the citric acid cycle that we'll look at in the next chapter. Succinate, one of the intermediates in the citric acid cycle, is transferring some hydrogens essentially to this coenzyme. The coenzyme is an electron carrier. And this is important for next chapter when we see what those electrons are going to do. But this reaction is catalyzed by an enzyme, succinate dehydrogenase. And again, dehydrogenase tells us we're removing hydrogens. It should be indicative of oxidation. But enzymes are still required to help most of these reactions. And that's going to be good later when we're looking at how do we regulate, the breakdown of sugars. So this is coenzyme A. Coenzyme A is derived from phosphorylated ADP and pantothenic acid. Now these are bonded by an amide bond to aminoethane thiol, which contains the SH reactive part of the molecule. So phosphorylated ADP, the phosphate group right here, pantothenic acid, and then amide bond to aminoethane thiol that has the functional part of coenzyme A. Coenzyme A is sometimes written as like this, because all we care about is the SH. We just want to see that SH part, and plus drawing the structure out is too long. So we'll usually show coenzyme A plus, something, and then we'll just link them. We'll link them together through the S bond. You'll see in a second. Actually, you'll see right here. So important functions of coenzyme A include the preparation of small acyl groups from larger molecules. So basically, we want these acyl groups so that we can make fats, and also there's another reason. We can anabolically build these into fats, but we can also break these two carbon groups down for energy. So we can oxidize these for energy. All right. Let's see. So coenzyme A's main functional wonder function is that it lets us make this molecule. Okay. Without coenzyme A, we wouldn't be able to make acetyl-CoA. Now, acetyl groups you're going to see are really common. Acetyl groups are the breakdown product of a lot of our molecules, but they're also used for building things. So you'll see acetyl groups a lot, in the next two chapters especially. But acetyl groups plus coenzyme A gives us acetyl-CoA. Okay. So acetyl-CoA is not the coenzyme, but acetyl-CoA is very important. So quick learning check. Go ahead and pause the video, give this a shot. See if you can match up the terms to their definitions. So carbon-oxygen bonds, NAD+. Reduced form should have an H in it, so 4. Transfer acetyl groups, coenzyme A. Contains riboflavin. Both of these guys contain riboflavin. One's just reduced and one's not. And then I'm missing something here. Coenzyme after C double bond O formation. So they're saying what is the form of NAD plus now that it's done this job? It's NADH. We don't really have to say there's also an H3O, because it has nothing to do with the form of this guy. But it's NADH. So when we are eating food, our digestion technically starts in the mouth, not just because we start breaking things down with our teeth, but we have enzymes in our saliva, right, that start to break stuff down. So once digestion begins in the mouth, it'll continue into the small intestine. So in the mouth, did I write this stuff down for you? Let's see. Let me stay here. So salivary amylase. This is just the enzyme amylase that's produced in the salivary glands. You have amylase produced in the small intestine as well. We wouldn't call that salivary amylase. This starts to break down polysaccharides into smaller polysaccharides. We call those dextrins. Dextrins are just smaller chunks of polysaccharide. But if we can break those down further, we can get maltose, which is a disaccharide, further even that into glucose. In the stomach, the partially digested starches will enter an acidic environment, and this low pH stops the carbohydrate digestion. It inactivates the enzyme. So that stuff doesn't break down anymore once it hits the stomach. Now, in the small intestine, things pick up again. The pH is about 8. Enzymes that are produced in the pancreas will hydrolyze those dextrins additionally, right, into more smaller chunks like di and monosaccharides. Enzymes of the mucosal cells in the small intestine will also hydrolyze maltose, lactose, and sucrose. So not just the disaccharide of glucose, but disaccharides that contain other monosaccharides as well, like galactose and fructose. Now, all of these monosaccharides then get absorbed. And once they're in the body, some of these other sugars, primarily fructose and galactose, will have to be converted into glucose because glucose is the primary energy source for muscle contractions, red blood cells, the brain, as it says there, but just all cellular respiration comes from glucose. And so, yeah, we can get energy from other molecules, but at the molecular level, it's glucose that our body runs on. And so we want to turn galactose and fructose into glucose as well. We're going to go through some of the reactions, and you'll see why glucose is so important. So, glycolysis. This is essentially the start of the breakdown, or the start of the catabolism. So, glucose in the bloodstream, right, enters our cells where it undergoes degradation in a pathway called glycolysis. Now, glycolysis, actually, I have what I was going to say right here. We'll come back to that in a second. Glycolysis is essentially going to take glucose from our diet. Oh, I guess it shows it here. Glucose from our diet and turn it into pyruvate. So that's glycolysis. Okay. Now, pyruvate will get converted into acetyl-CoA. Okay, which is, remember, the coenzyme A with an acetyl group. Acetyl-CoA will then enter the citric acid cycle, and we'll look at that in the next chapter. The citric acid cycle will produce many of the reduced forms of the other coenzymes that will then go on to the electron transport chain. So glycolysis just refers to essentially taking a 6-carbon molecule and turning it into 2 3-carbon molecules. Okay. So it takes place in the cytosol. This is separate from like where DNA replication and transcription take place, right, in the nucleus. Glucose is the product that enters glycolysis, and we're going to break it essentially in half. We're going to take the 6 carbons and turn them into 2 3-carbon molecules. Now, there are some classifications of the reactions. In the first, there's 10 reactions. In the first 5 reactions, we have to put energy in. Our molecules aren't ready to enter into glycolysis and to give us energy, so we have to prep them a little bit. So energy is first required, and we in the form of adding phosphate groups. So glucose here you can see, now this is just a diagram. We'll go through each one independently, but just to kind of give you an idea here, we've got the 6 carbons, right? We're going to bring in an ATP hydrolyze it into ADP, and essentially one of those phosphates is going to add to our glucose, glucose 6-phosphate. Glucose 6-phosphate will get converted into fructose 6-phosphate. So we know that that just changes the aldose into a ketose, right? Fructose 6-phosphate then will also get prepped ATP into an ADP, adding a second phosphate group onto our molecule, so we'll get fructose 1,6-bisphosphate, and then that molecule will get spliced in half. And we won't end up with two of the same molecule, right? We're going to get two different molecules. We're going to get dihydroxyacetone phosphate and glyceraldehyde 3-phosphate. Now, we like glyceraldehyde 3-phosphate. So, what you'll kind of see here, and you would see it better if I hadn't sort of erased it, but what you'll see here is that we're going to convert the dihydroxyacetone phosphate into glyceraldehyde 3-phosphate. So essentially we're going to have two of these glyceraldehyde 3-phosphate molecules. Now, in reactions 6 through 10, we get to get energy back. We're going to get the payoff here. So in the first part of these steps, you'll see here, energy is going to be obtained from hydrolysis of these energy-rich compounds. So those two glyceraldehyde 6-phosphates, glyceraldehyde 3-phosphates, sorry. Those glyceraldehyde 3-phosphates are going to get turned into 1,3-bisphosphoglycerate. Now, let's see, how does this happen? It looks like two phosphates and two NAD pluses are required. They essentially get reduced to NADHs, and it looks like they add another phosphate on here. Okay, so that doesn't cost or so that isn't an energy releasing step or an energy gaining step. That's just another step. So those two phosphates that are floating around from being hydrolyzed earlier, they essentially get added onto our molecules. So our molecules have two phosphates on them again. So that's one of the steps where NAD was required. And then after that, it looks like they go from having two phosphates to only having one phosphate. So the best way to say it is we're able to turn an ADP into an ATP. So we've already made back, right, some of the energy that we put in. And if you remember, let's go back real quick. We only spent one ATP, two ATPs to make this happen. So we're two in. And in this step, we just got two back out. This was two ADPs into two ATPs. So now we're even. 3-phosphoglycerate, which is the product of 1,3-bisphosphoglycerate losing one of its phosphates, now gets sort of rearranged. So 3-phosphoglycerate becomes 2-phosphoglycerate. And then some waters come in here. Now remember, at all of these steps, we have two of everything. That's why everything's doubled, because we have times 2 times 2, remember. So we have two of these 2-phosphoglycerates. We're going to, looks like, use some water here to do some rearranging, and we're going to take 2-phosphoglycerate and make phosphoenol pyruvate. Phosphoenol pyruvate is another high energy molecule. And this high energy molecule is going to be able to be used to make two more ATPs. And since we've gained 4 and we put in 2, there's a net gain of 2 ATP molecules. So it says here 4 were synthesized, but remember we put 2 in. So that means this process of just turning glucose into some pyruvates, so 6 carbons into 2 3-carbon molecules, generates 2 ATPs. 2 ATPs that we didn't have before. We put 2 in, we get 4 back. So it makes energy. It's not a lot, but it makes energy. And we're going to see that smaller organisms that aren't able to do electron transport like we can, they survive on this energy. All right, let's look at those reactions in a little more detail. All right, So first we see the phosphate group actually being added onto our glucose to make glucose 6-phosphate. The enzyme hexokinase does this job. Reaction 2 is an isomerization. So glucose 6-phosphate, which was an aldose, becomes fructose 6-phosphate, which is a ketose. And you can see here kind of reflected in the way that the ring is formed, right? We've got a 6-membered ring going into a 5-membered ring. So it looks like fructose. Fructose 6-phosphate. This was catalyzed by phosphoglucose isomerase. In reaction 3, we get another phosphorylation. Here, another ATP provides a second group. We have another kinase. Kinases are often responsible for phosphorylating things. This one's called phosphofructokinase, and it transfers another phosphate group onto the molecule. So this was the second ATP we had to put in to this reaction. And you can see 2 phosphate groups now. Fructose 1,6 bisphosphate. Cleavage step. So here, fructose 1,6 bisphosphate, it gets split into those 2 phosphate isomers, glyceraldehyde 3-phosphate and the dihydroxyacetone phosphate. Now, aldolase is responsible for producing those two molecules. Now we're going to need to convert this one into that one. So reaction 5, isomerization, the dihydroxyacetone phosphate is catalyzed by the enzyme triose phosphate isomerase. Triose meaning 3 sugars, phosphate isomerase meaning it's going to move the phosphate to make an isomer. So the second molecule of glyceraldehyde 3-phosphate then is produced. So now we have 2 of those, and all 6 carbon atoms, like it says, have been reduced into 2 identical trioses. So trioses move on to reaction 6. The aldehyde group of glyceraldehyde phosphate, or 3-phosphate, is oxidized to a carboxyl group, and it's oxidized by NAD, which makes NADH. A new phosphate group is transferred to each of these carboxyl groups, forming 1,3-bisphosphoglycerate. From there, 1,3-bisphosphoglycerate transfers one of its ATP molecules, or phosphates, sorry, to an ADP molecule by phosphoglycerate kinase. And so since there was 2 molecules and 2 phosphates and 2 ADPs, we make 2 ATPs. So an enzyme transfers one of those phosphates off. Reaction 8 is isomerization. So the 2 molecules that are 3 phosphoglycerates end up becoming 2 phosphoglycerates. So an enzyme called phosphoglycerate mutase moves the phosphate group from the number 3 carbon to the number 2 carbon. So now we have 2 molecules of 2-phosphoglycerate. A dehydration step, catalyzed by an enzyme called enolase, basically removes some water. Now this enolase can add water or remove water, and in this case it's removing a water. And so then we get from there, a loss of an H and an OH. We get 2 of these high-energy phosphoenol pyruvate molecules. So again, times 2. And this molecule here has a phosphate on it that we're going to use to make another round of ATPs. So the phosphate group moves from phosphoenol pyruvate to the ADP molecules by pyruvate kinase, once again. And, a kinase, in this case it's pyruvate kinase, because the product here is going to be our pyruvate. And again, it's the 4th kinase enzyme. So if you go back and look at all of those different kinases, there was the one that made glucose 6-phosphate, the one that made fructose 1,6 bisphosphate, the one that transferred, the glyceraldehyde 3-phosphates, and so on. So there we go. So now we have our pyruvate molecule, and you can see it's a 3-carbon molecule, and again we have 2 of those. And those are going to move on in the next chapter. So here, we'll take a look a few things. First, just this glycolysis series of reactions, and then we'll talk about what some organisms do with some of these products. So um, let's not forget actually that okay, no, full reaction here. So from our glucose and two NAD pluses and two ADPs and two inorganic phosphates, we can get two pyruvates, two NADHs, two ATPs, and so this is net, two net ATPs, and then some waters and hydrogen ions. So this would be the net reaction, because of course we know that um, it costs some ATPs also, right? And we really made four, but so there's our overall reaction. So from every glucose we get two pyruvates, two NADHs, and two ATPs. Now there are other sugars that are important. Fructose and glucose we mentioned. Those have to enter glycolysis after they get converted into intermediates. So in the muscles and the kidneys, fructose gets phosphorylated into fructose 6-phosphate, which we know can enter glycolysis in reaction 3, because glucose gets turned into fructose 6-phosphate. So fructose can enter in that step. Um, Fructose in the liver gets converted into fructose 1-phosphate, which can be converted into uh, 2-glyceraldehyde 3-phosphate molecules via a different series of reactions. They're not showing those reactions here, but then it can enter glycolysis at that step. Galactose can react with ATP to yield galactose 1-phosphate, which can be converted into glucose 6-phosphate. And so that one can enter at reaction number 2. So these other sugars you can see can be converted into the different intermediates so that they can enter glycolysis. They don't have to necessarily be converted into glucose, so that they can enter up here. They can enter wherever we can get them, wherever we can get them into the cycle. It's regulated, glycolysis, by 3 enzymes. So remember our enzyme chapter and how we regulate enzymes. In reaction 1, hexokinase, which is the one that first puts the phosphate onto the glucose, it's actually inhibited by high levels of glucose 6-phosphate. So if you already have lots of glucose 6-phosphate, that blocks that enzyme from continuing to make more. Reaction 3, phosphofructokinase, this is the one that adds the second molecule of phosphate onto those enzymes, or onto those, this is the one that makes fructose 1,6-bisphosphate. It's also an allosteric enzyme. So high levels of ATP, so if you already have lots of ATP around from presumably glycolysis, citric acid cycle, and electron transport, if we already have energy, then they would turn off this enzyme. But also high levels of ADP and AMP would turn this on. So if you were metabolically active and you use a lot of ATP and there was a lot of these guys floating around, they're going to stimulate phosphofructokinase to start, making more of that glycolysis product. Reaction 10, pyruvate kinase. This one's inhibited, again, by high levels of ATP or acetyl-CoA. And that one may not make sense right away, but pyruvate, we're going to see in the step before the citric acid cycle gets converted into acetyl-CoA. And so if you have a lot of acetyl-CoA, it's probably because you had already a lot of pyruvate, so you don't need to make more pyruvate. So these are the 3 sort of control points in that reaction. Now there is a pentose phosphate pathway. This is a different pathway for glucose. So glucose wouldn't be oxidized for energy going into this pathway. It would actually be used to both produce the coenzyme NADPH and 5-carbon pentoses. So NADPH would be used for anabolism, protein synthesis or lipid synthesis or something, or nucleotide synthesis. The 5-carbon pentoses would also be used for nucleotides. Right? We need those pentose sugars for our to make ribose, so we can have DNA and RNA. So the NADPH, the reduced form of it, NADP plus, this is an important coenzyme in anabolic pathways, including, like I said, fatty acids and nucleic acids, but also cholesterol. Um, uh, like we talked about, cholesterol has a lot of functions too, and it's a precursor for a lot of steroid molecules. So NADP, another important coenzyme. But again, it's more utilized, or it's produced as part of the phosphate pathway and not part of glycolysis. In glycolysis, we're using NAD plus and we're making NADH. So pyruvate. What does pyruvate do? Well, now that pyruvate has been made from glucose, it can enter pathways that will continue to extract energy. We're going to look at what happens in a few scenarios. If the available pathway has oxygen, if there's oxygen, then we call that aerobic, and oxygen is going to be able to help us convert pyruvate into acetyl coenzyme A, or acetyl CoA, and carbon dioxide. So that's already going to be breathing out, essentially, right? Your 3-carbon pyruvate to a 2-carbon acetyl CoA, one of your carbons just got breathed out. If you do nothing but breathe overnight, then you lose weight because your carbon atoms are flowing out of you, right? Every time you breathe in some oxygens, you're breathing them out a little heavier with a carbon on them. So when oxygen levels are low, and we would consider that anaerobic, pyruvate has to be reduced to lactate. And so we're going to look at these. So in aerobic conditions, lots of oxygen present, we're going to convert pyruvate into acetyl-CoA, and this will then go into the mitochondria so that it can be oxidized further. The NADH will go to the citric acid cycle to be, which is also in the mitochondria. Sorry. Acetyl-CoA goes on to the citric acid cycle, which is in the cytosol. The NADH goes into the mitochondrial matrix to be part of the electron transport chain. So that's in the mitochondria. Now, there are some parts of the citric acid cycle that do go in and out of the mitochondria, but for the most part, let's just say things take place in the cytosol, and then some parts move to the mitochondria, like the electron carriers. Excuse me. So pyruvate gets oxidized when the carbon atom is removed as CO2 and coenzyme NAD is reduced. And so this is an additional step, right? This is what happens to pyruvate after we've exited glycolysis. So another coenzyme A, actually the first coenzyme A, and another NAD+. So later, when we are adding up all of the NADHs that were formed, we're going to want to do that so that we can figure out how much energy total we've made. We've already seen ATP can form directly in the cytosol, but most of our ATP is going to come from a process called oxidative phosphorylation that takes place in the mitochondria as part of the electron transport chain. And that involves heavily the NADHs and the FADH2s that we make. So when we go back later to add up all of the NADHs that we made, let's not forget that we did form some in glycolysis, and we're also forming some right here in this reaction. This reaction that takes pyruvate and turns it into acetyl-CoA. And then acetyl-CoA will continue into the citric acid cycle, and we're going to end up using even more NAD pluses in that scenario. Now, in anaerobic conditions without oxygen, pyruvate has to be reduced to lactate and NAD plus. Now, so what's happened here, okay? Instead of being able to take pyruvate and add another NAD plus and add another coenzyme A in order to continue on, the cycle, there is no oxygen. So what happens is NADH builds up, and eventually all of the NAD plus is gone. We don't have any more. And if we don't have any more NAD plus, then we can no longer continue to use NAD plus in glycolysis. Glycolysis would actually shut down, right? Remember, pyruvate is what happens after glycolysis. So glycolysis finishes, we have pyruvate, but if glycolysis stops at any point because we run out of NADH or NAD plus, then we're not going to have pyruvate anymore. So we want to keep this process going. And so the only thing to do is to keep regenerating NAD plus. And the only way to do that is to reduce or to oxidize NADH again. So this is what happens in your muscles when you are very active. If you're not breathing well, if you don't have good circulation, you start to be a little bit oxygen deprived in your muscles. So instead of the pyruvate continuing to be converted into acetyl-CoA or acetyl-CoA so it can go and make energy, it actually, your muscles make the choice. We need to start oxidizing oxidizing NADH back into NAD plus so that we can keep glycolysis going. And the expense of that is turning pyruvate into lactate, which can become lactic acid, right, in your muscles. They say that's what gives you the burn. Now it's been debated whether or not that burn is there to slow you down or make you go harder. They showed in a study with mice or rats or something, when mice couldn't feel the burn in their muscles, they didn't go harder. They didn't try harder. They almost slowed down. It was like counterintuitive. So they think maybe that sensation is there to push you, to keep you going or something. But anyways, what happens is, right, we don't have oxygen present, so that pathway for pyruvate plus acetyl-CoA shuts down, and instead, the only way we can do something with our NADH instead of sending it to the electron transport chain is to just regenerate NAD plus, because at least if we can keep glycolysis running, that always gets us 2 ATPs every time. 2 ATP, 2 ATP, 2 ATP. We can definitely get more from pyruvate, but only when that pathway is open. And if that pathway is not open, then the pyruvate does us no good, and the NADH does us no good. So rather than let it build up and have everything shut down, we can regenerate it to keep glycolysis going. At least we'll get some ATP. At least we'll get some. All right. Now, so you can see here, accumulation of lactate causes the muscles to become tired. I mentioned, at least we can get some ATP, even though we're making lactate, which we don't really want. Now, some organisms don't make lactate. For example, I'll show you here some of the pathways. So pyruvate in aerobic conditions can go on to become acetyl-CoA. And then, of course, that goes into the citric acid cycle. The NADH goes on to electron transport chain. And again, all of the other coenzymes that we'll make here, more NADHs, more FADH2s, they go to electron transport chain. And this is just more and more and more ATP. But if that's not available, then we have two options. One, make lactate, just so that we can have more NAD+, so that at least the beginning part, or at least this can still happen, right? We need this to keep happening. So NAD+ just goes back up to the top and lets pyruvate keep being made. Now, some organisms will actually ferment. So what they do is, rather than make lactate from pyruvate, they will go and convert pyruvate into acetaldehyde, and then acetaldehyde into ethanol. And that process also regenerates the NAD+, so that they can at least send that back up to the top and keep glycolysis going, because there's nowhere else for the NADH to go. And of course, ethanol is that product that we like from all of the fermentation that yeast does, right? So here's that reaction. So pyruvate decarboxylase removes a carbon dioxide from pyruvate. Um, this might be part of the, the CO2 that forms during the conversion of the ethanol to um, what's it called? The ethanol to sorry, pyruvate to ethanol. I was reading ethanol, going, why am I saying this word wrong? My bad. The pyruvate into the ethanol. At some point in my mead making, I think right after this step was happening, I had a leaky vessel and oxygen was seeping in. And instead of the oxygen, so what was happening was instead of anaerobic conditions that should have driven the ethanol into ethanol, I was getting a lot of this acetaldehyde becoming acetic acid. And it was giving it a very vinegar-ish smell. And so I had to quickly purge the oxygen, maintain a good CO2 pressure so that that wouldn't keep happening. I don't know if I've saved my mead. It does still taste kind of funny. But I was able to figure out what happened and why I wasn't getting a lot of ethanol and why I was getting a sort of soury taste to it. But it makes sense. It makes sense. So that's fermentation, of course. Depending on what you ferment, you get different products, right? Barley makes beer, grapes is wine, potatoes, vodka, rice, sake, corn or rye, your whiskeys, honey, of course, for my meads. Yeah, okay. Let's talk about glycogen synthesis and degradation. So here you can see, this is kind of a bad thing that's happening here. This shows excess stored glycogen in blue in a person's liver. So they've got too much glycogen here because of something called Cory's disease. I don't know if that has to do with the Cory cycle. We'll look at that later. But essentially, right, if you have a lot of glucose, more glucose than you need to be putting into to glycolysis, you can store it. So the excess glucose gets stored as glycogen, and it gets synthesized by your muscles and your liver. They store it. There's enzymes involved. We'll look at those. So we know that glycogen is a polymer of glucose that has alpha 1,4 and branches alpha 1,6 linkages. It's formed when there are high levels of glucose 6-phosphate. So if glycolysis is making lots and lots of this glucose 6-phosphate, this stuff's ripe for storing. It's not formed when our glycogen stores are full. And so if we have extra glucose on top of glycogen, then that glucose gets converted into triglycerides and stored as fat. Glycogenesis is what we call the process of forming glycogen. And it again produces glucose 6-phosphate in reaction 1 of glycolysis. That glucose 6-phosphate then gets converted into glucose 1-phosphate. So the phosphate group moves from the number 6 carbon to the number 1 carbon. An enzyme phosphoglucomutase does that, and it utilizes UTP instead of ATP in this reaction. Reaction 2, we get glucose 1-phosphate being activated, I guess activated by the UTP. Glucose 6-phosphate gets activated, yeah, and so you can see here it's been activated by an additional phosphate group. So the UTP became UDP, but then because there was already a phosphate here, now we have 2 phosphates, it's UDP glucose. Oh, my bad. The uridine actually stays on here. So we didn't do that. We basically had a UTP and then 2 phosphates came off and the UMP stuck on here. And because there was already a phosphate, it's UDP. Okay, so it's activated with the addition of this uracil group. And now this high energy compound, now we have UDP glucose and pyrophosphate, which is what they call this, this leftover of the UTP. So reaction 3 is the actual synthesis part. So now we've got these activated glucoses. Now the glycogen synthase catalyzes the breaking of the phosphate bond to the glucose. So it's going to catalyze this bond breaking. And then when that glucose is released, an alpha-1,4 glycosidic link forms with the end of a glycogen chain. So it's going to add on to this side. And we'll get a free one of these. And so they grow this way. All right, so this enzyme, right, it's activated by insulin. So when insulin's around, this thing will go and actively store extra glucose that you have as glycogen. Glucagon and epinephrine, which we haven't talked about, are inhibitors of this particular enzyme. I believe glucagon is, actually I don't remember what glucagon is. So glycogenolysis is the opposite of that, sort of. This is when you want to break down glycogen, so glycogenolysis. The first reaction is the phosphorylasis. Sorry. Glucose molecules get removed from the glycogen chain. Glucose molecules get phosphorylated and they get turned into glucose 1-phosphate. So as they are cut off from here one by one, and again, this molecule, glucagon, okay, so here we go. Insulin promotes the forming of glycogen, and glucagon promotes the lysis of glycogen. And so that's why they are inhibitors of the corresponding opposite enzymes. Whatever enzyme is activated by insulin is inhibited by glucagon, and vice versa. So in reaction 2, we get hydrolysis of the branches. Glycogen phosphorylase cleaves the alpha-1,4 bonds, and the debranching enzyme cleaves the 1,6 bonds, so that those now branches can just be broken down through the reaction 1, the phosphorylasis. Because they're all connected the same, right? We just have to deal with those branches, and then they get broken down one by one. Reaction 3, glucose 1-phosphate gets converted into glucose 6-phosphate so that it can enter into glycolysis. So if you are in need of energy, your glycogen breaks down and it gives you an already activated glucose to go right into glycolysis. Reaction 4, dephosphorylation. Cells in the liver and kidney have a glucose 6-phosphatase that will actually hydrolyze glucose 6-phosphate to yield free glucose. So this will put free glucose into the bloodstream so it can float around. So this is not even glucose that's ready for glycolysis. This is just bloodstream glucose. Obviously, so that it can, remember, glycolysis is inside the cells. So bloodstream is getting it to other cells. So remember, it's stored in the liver and the kidney, right? So this is how it gets made into the right form so that it can travel to all the different cells, like your brain, where it'll get absorbed and then, turned back into glucose 6-phosphate so it can undergo glycolysis inside that cell. Okay, it doesn't float around the body or in the blood as glucose 6-phosphate. All right, quick learning check. Give it a pause. See if you can make sense of glycogenesis or glycogenolysis. So activated by low levels of blood glucose, that would be genolysis. Converts glucose 1-phosphate into glucose 6, genolysis. Activated by high levels of glucose 6-phosphate is glycogenesis. And then the galactose and the UTP. We saw this when we are activating our glucoses so that they can be added onto the chains and grown into glycogen, right? Glycogenesis. Couple more terms, not that many. Gluconeogenesis is when we make glucose from a lack of glucose. So glucose is synthesized in the tissues of the liver and in the kidneys. Tissues that use glucose as their main energy source are the brain, the muscles, the red blood cells, and so on. And so we are actually able to make glucose so that we can feed some of these important organs. So glucose is the primary energy source of the brain, as we mentioned, and a deficiency in that can actually impair that function. So when our glycogen stores get depleted, which happens typically after about a day, the liver cells can make more glucose via gluconeogenesis. Glucose is synthesized in the cytosol of the liver cells and some in the kidneys. Glucose can supply us with, glycogen, only about a day's requirement of glucose. So if you fast for a whole day, you've consumed all of your glycogen. And we know that that's made of glucose. So most of which is synthesized in the cytosol of the liver cells. So to begin gluconeogenesis, we have to take carbon atoms that didn't come from sugar, because if we had sugar, we wouldn't need to make glucose. So these are like amino acids, these are like fats. And they can get converted into pyruvate, right? Pyruvate, we know, is the end product of glycolysis. So glucose can be turned into pyruvate. So it makes sense then that if we can turn glucose into pyruvate, we can turn pyruvate back into glucose. Now, it's not exactly a repeat of the same reactions, though some of them are. You can see here in the beginning, there's a couple of different steps, but then once we're at phosphoenol pyruvate, we can actually just reverse the direction of a few of these reactions. The fructose 1,6-bisphosphate, different enzyme to get to the fructose 6-phosphate as opposed to the other direction. And then again, the glucose 6-phosphate can be made into glucose via a different enzyme than the one that sort of originally turned glucose into glucose 6-phosphate. But aside from a few different steps, it's a lot of the same recycled molecules, the same enzymes that can go both ways. So that's really convenient for us. And we'll look more into this when we start looking at fats and amino acids in the diet. Here we're just vaguely saying, we can make them from carbon sources. I'll show you which carbon sources specifically we can use to make sugars in the next, in chapter 24. But here just know that carbons can be turned into pyruvate, some of them, and those can be turned into glucose. So we have, from glucose, we can make other sugars, specifically for nucleotides via the pentose pathway. We can make glycogen via glycogenolysis, and we can also, via glycogenesis. I should have drawn my arrow like this. And then we can break it down via glycogenolysis. We can break down glucose for energy into pyruvate via glycolysis or glycolysis, but we can also, from pyruvate, make glucose via gluconeogenesis. From pyruvate, we talked about the fact that we can oxidize this molecule into lactate as a way, reduce this into lactate as a way to regenerate NAD+. We can also continue oxidizing it in the presence of oxygen into acetyl-CoA to move on to the next chapter, or some organisms can reduce lactate into ethanol, again, to produce NAD+ again, to be able to make glycolysis continue happening. We didn't talk about the Cori cycle, but lactate can be converted into pyruvate. Pretty sure we didn't look at that at all, though. Okay, I think that's about it. So one more quick learning check, and then that's it. So see if you can pair the right words to the right descriptions here. Pause it, give it a shot. Gluconeogenesis, glycogenolysis, glycolysis, and glycogenesis. The name is in there, glycogen. Glycogen. So look for that. Glyco. I wish they wouldn't mix things up like that, but genesis means to make. So to make glucose. To make glycogen, to make glucose. Neogenesis maybe here means to make a new glucose molecule. I don't know. Trying to see if I can help. All right, that's it. So email me if you have questions. See you in the next one.